Hi, welcome to Tuesday at 5 o'clock. So today we are going to be walking through this AQA exam, paper 2 from the AS syllabus and from the year 2018. So welcome to those of you in not live stream land, because you're going to have the added bonus of being able to stop the video so that you can have a go at the questions possibly before I put up my answers and go through the mark scheme. You'll notice that I do not write exactly what is in the mark scheme for every question, because I have been quite careful in trying to complete this paper without looking at the mark scheme beforehand. And therefore, I'm writing as I would think. And I don't get every single mark. I get an A star, but I don't get every single mark. And so I think that's worth you noting. The other thing that I do is that I'm going to show you exactly where I've earned the marks. So where I put a tick and I write the mark point number, that's where I've put that tick is exactly where that mark point has been earned. So let's get going, because this is going to take a little bit of time, but I'll try and do it as efficient as I can. So here we have a list of structures, A to E, and we have got to put in the letter depending according to the statement that is describing the structure. So has stacked membranes arranged in parallel and contains DNA. I'm thinking chloroplast, therefore B. It's made of a polysaccharide, therefore a cell wall. It's an organelle and not surrounded by two membranes. Well, the only one on this list is E, the Golgi apparatus. Mitochondrion, nucleus, and chloroplasts all have double membranes. And fortunately, I got those ones right. Human breast milk is produced and secreted by gland cells. Any gland is just an organ that secretes something. So the pancreas, for example, is a gland that secretes insulin. These gland cells have adaptations that include many mitochondria and many Golgi. The milk contains a high concentration of protein. So these gland cells are the things that are producing the milk. And the milk contains a high concentration of protein. So we're kind of assuming that the vesicles that, notice that we're talking about Golgi vesicles, not the Golgi apparatus itself. So here we're talking about those vesicles that come off the Golgi apparatus. So explain the role of these cell adaptations in the production and secretion of breast milk. So here, the mitochondria are going to be producing ATP so that bigger molecules, proteins, can be made up from smaller ones. You might remember that that is one of the main purposes of the energy that's released from respiration that you learnt about at GCSE. And then the other thing is that the Golgi vesicles, also, they are the things that carry the modified proteins from the Golgi apparatus to the cell surface membrane, where those vesicles fuse with the cell surface membrane to secrete those modified proteins into the milk. So this is what I wrote. Here is the mark scheme. And here are the examiner's little notes. And therefore, this is where I got the mark point 1. So that, sorry, I forgot that this is what I said. It talked about the ATP being used to move the vesicles. Notice that synthesis of protein is also another one. And that the Golgi vesicles transport the protein, or the modified protein. That's why you've got glycoprotein here. Okay. Oh, I should say, if you've got any questions, do type them in the chat. Really happy to answer any as we go along. I'm probably running about 10 seconds be ahead of what you are seeing. So just be aware that there's that little bit of delay. And the other thing that I am really supposed to say, and I hate saying, because I'm not a marketeer, I'm not a salesperson, But if this stuff is useful, do subscribe to the YouTube channel because I am posting new videos, new live streams every week. Anyway, let's get on with what we're here for. 2.1, describe how a peptide bond is formed between two amino acids to form a dipeptide. Well, the first thing that comes into mind is that the term condensation is going to be in that answer somewhere. But then I'm kind of thinking, okay, once I've said that condensation and that releases water, what else am I supposed to say? So now I'm thinking about those two amino acids joining, and I realize that I can put another bit of detail, that it occurs between the amine group of one amino acid and the carboxyl group or carboxylic acid group of the other amino acid. That was literally my thought process as I was writing this answer. So I only thought of this second bit as I was writing the first bit. And then by the time I'd got to the end of that rather long sentence, I kind of thought that I'd probably written enough to get those 2 marks. And I had, fortunately. So mark point 1 for condensation, mark point 2 for talking about the 2 groups. The secondary structure of a polypeptide is produced by bonds between amino acids. Describe how. Here we've got to make sure that we're identifying the level of structure that they want us to. This is the secondary structure. Primary structure, order of the amino acids. Secondary structure, those alpha helices or the beta pleated sheets. Tertiary structure is the actual 3D shape of the protein. And quaternary is when you have more than one polypeptide joining to make the protein. So I know that the secondary structure is as a result of hydrogen bonds, and that is between the amine, oh sorry, no, that's not true, between the NH group and the CO groups on either side of the peptide bond. If you imagine it, you've got the NH group coming up, you've got the CO group below, and what happens is that as that alpha helix is produced, all that beta pleated sheet, the H of one of the NH group forms a hydrogen bond with the CO of a non-neighboring amino acid. And that is what stabilizes the secondary structure. So I got the mark for hydrogen bonds, and I got the mark for saying where it occurred. Silly old me, I actually think this is almost the easier thing to write, this phrase here. The fact that alpha helices and beta pleated sheets are formed, I didn't even think about it because I was so fixed on describing the hydrogen bonds. 2.3, 2 proteins have the same number and type of amino acids, but different tertiary structures. Why on earth would that be? Well, that's because they may have the same number and the same type of amino acids, but those amino acids are in an utterly different order between those 2 proteins. And therefore, as a direct result of that, there would be different ionic, different hydrogen bonds, and different hydrophobic interactions that would occur between non-neighboring amino acids, which would stabilize the tertiary structure. Oh, sorry, I should have also said, of course, disulfide bonds. That was the other. Actually, it's more obvious a bond to talk about for the AQA syllabus than the hydrophobic interactions. So here's the mark scheme, different sequence of amino acids or a different order, and the bonds. There we go. Oh, this is just in case the candidate assumes the proteins are the same. I can't envisage that, but maybe that's just me being a bit teachery. So mark point 1 and mark point 2 gained. 3.1, describe the relationship between the size and surface area to volume ratio of organisms. So hopefully this is relatively straightforward. that as the size of an organism increases, the surface area to volume ratio of that organism decreases. As organisms get bigger, they have relatively less surface area in comparison to the volume, that 3D volume that they take up. I always use the analogy, think about which you'd be most concerned about getting cold, a baby or an adult. It would be the baby, because the baby has got a far greater surface area in comparison to the volume that they take up than the adult. And that's why the baby will lose heat far more quickly than an adult. Anyway, moving on. Scientists calculated the surface area of a large number of frogs' eggs. He found that the mean surface area was 9.73 millimeters squared. Frogs' eggs are spherical. So we've got this mean surface area for a frog's egg. And they were very kindly, they've given the equation for it. Use this equation to calculate the mean diameter of a frog egg. So, oh, sorry, I've just realised that I haven't done this, silly old me. There we go, I got the mark. Onto this question. So, we are told that 9.73 equals 4 times pi, and I happen to use 3.142, times by r squared. So does it make sense that r squared is going to be 9.73 divided by 4 and then divided again by 3.142? In other words, this. And if we want r, we need to square root that to get r. But then we need to remember that the diameter is 2 lots of r. And therefore, the diameter is 1.76 millimeters, which is what they were happy with. And you're given a range because it depends what value of pi you use. And here is the gubbins that gives you lots of other ways of getting a mark if you didn't get the right answer. Okay, 3.1. Sorry, wrong, let me click. Here we go. The scientists calculated the ratio of surface area to mass for eggs, tadpoles, and frogs. He also determined the mean rate of oxygen uptake by tadpoles and frogs. The scientists used micromoles per gram per hour for the rate of oxygen uptake. And what they're saying here is that this would be micromoles of oxygen per gram of the egg per hour of time, obviously. Suggests why he used micromoles in these units. And the most obvious thing that I thought of is that it's just a really tiny amount of oxygen that is being taken up in that period of time. So that's what I put, and that's what gave me the mark here. I didn't. This is obviously also true. If you're talking about moles, you'd be having multiple decimal places rather than just making the unit micromoles. So, you know, that's perfectly reasonable, but I didn't think of it. The scientists decided to use the ratio of surface area to mass rather than the ratio of surface area to volume, and he made this decision for practical purposes. Suggest one practical advantage of measuring the mass of frogs' eggs, tadpoles, and adults compared with measuring their volumes. Now, I went down the route of just kind of going, hey, all you've got to do to measure a mass is to put the thing on a balance. Whereas if you're measuring a volume, you've got to do a calculation of some kind. And I just thought that that sounded much simpler. Didn't get me the mark though. So look, I wasn't convinced that this last mark point, that I actually quite nailed it. I wasn't completely sure. I haven't given myself the mark. But notice that this is the other one that I thought was kind of reasonably obvious, that if you think about, you know, the shape of an egg is nice and spherical, but if you think about the shape of a tadpole and the shape of an adult frog, oh my goodness, trying to actually work out the volume of those things, quite tricky. So, you know, but I didn't give myself the mark for that. Hi, can you take a little pause before answering the questions so that we can have time to attempt them? Well, the trouble is that I will be here until 8 or 9 o'clock if I try and do that. So I am literally making this as an efficient live stream as I possibly can. Because of course, this exam would take an hour and a half for you to do. So, you know, it's obviously going to take you a period of time to think about the question and to at least even have a go in your mind as to what the answer is. I think it's just going to make the live stream too long and too slow. I do appreciate it. You might actually be better off watching it when it's just automatically uploaded to my YouTube channel so that you can stop the video when you're not watching it in live stream land. I hope that makes sense. So explain why oxygen uptake is a measure of metabolic rate in organisms. Now, I have to say, I think this is a bit of a weird question because, you know, kind of go, well, why is oxygen uptake a measure of metabolic rate? Metabolic rate is just all of the reactions that occur in the cell, whatever kind of reactions they are. So why is oxygen uptake a measure of metabolic rate? And I kind of thought, you know, well, respiration is such an important reaction that I just kind of thought, well, you know, maybe we can say that that is representative of all of the reactions that are going on in the cell. And therefore, oxygen uptake can be measured as an estimate of the metabolic rate in organisms. That was the main way my mind works. Walid, also probably worth saying that, you know, when I'm doing all of this bit of blurb, as I'm talking about how I'm thinking about it, it may also be the time when you can kind of be going, well, that wasn't the way my mind worked. This is what I would have written, you know, just a thought. Well, Brooke, my live streams start at 5. So that's pretty standard every week, but I am taking a break as of next week because I'm going on holiday. That's quite exciting. I've put a little message up on my community tab so that you know, my dear old subscribers know what I'm doing. So I won't be around for 3 weeks after this week. So anyway, so here is what I wrote. Because oxygen is required for aerobic respiration, which is a representative chemical reaction. So here is what, here is what the mark scheme says. Do they give me? Okay, the examiners gave a little bit of guidance here. Notice you can never, ever use the words produces energy or makes energy, because thermodynamics say that energy can neither be created nor destroyed. You can never make it, you can never produce it. It can be released. Worth noting though, you can always say that ATP is produced because ATP is a chemical. So you can say that, that is correct. So I think, did I give myself the mark? Oh, I did give myself the mark on that one. A student who looked at these results said that they could not make a conclusion about the relationship between the stage of development and the metabolic rate. Use the information in table 2 here to say why they were unable to make a conclusion. Well, the really obvious thing to say, and this is 3 marks, tricky. The really obvious thing to say was that you've got no information about the egg. So I kind of wrote that and I kind of thought, well, that's 1 mark. And then I sat there for a little while thinking, well, what else can I say? because it is a mean rate. They've obviously, let's assume that they've taken a reasonable number of tadpoles and adults to come up with these figures here. And then I thought, of course, at AS, you learn about statistical tests and you learn about the use of standard deviations when you take a whole collection of data and calculate the mean. And it did require my brain to kind of jump a bit. And it was a bit of a stretch because even I, as a teacher, always think of all of those statistical tests almost as real year 13 work. But at AS, you are expected to know the 3 statistical tests and the type of data that they can be used on. Not very much else. You're not expected to know about p-values, not expected to know about interpreting them, but you are expected to know that stuff. So it was, therefore, this is the answer that I wrote. Had no data for the egg. Stop for a few seconds. No standard deviations. And as I was writing standard deviations, I could have just written all statistical tests, but then I clicked, we are comparing means. We are seeing whether 2 means are different enough. And therefore, instead of just saying statistical tests, I could actually be more specific and say, or the student t-test were calculated on the 2 other means. So that's what I wrote. And here are the mark points. Okay, here's the examiner's guidance. Mark point 1, I didn't get mark point 3. I only got, sorry, that should say mark point 3. That's not mark point 2, it's mark point 3. So I got 2 out of the 3 marks there. Give 2 similarities in the movement of substances by diffusion and by osmosis. Well, a really obvious one is that they both substances, both whatever the substance is moving by diffusion and water, which is the only thing that is moving if you're talking about osmosis, both of them go down their concentration gradients. The substance in the case of diffusion and water in the case of osmosis. Because water is the diffusion, osmosis is the diffusion of water. And it goes from a higher water concentration to a lower water concentration. And the correct term for that is higher water potential to lower water potential. And the other thing is that both of these are passive. So here is what I wrote. Here is the mark scheme. Here are the examiner's guidance. Notice you have got to say down a concentration gradient. You cannot say along it or across it. You either say down the concentration gradient or against the concentration gradient. Those are the terms to use. So, yes, I got mark point 1 here, and I got mark point 2 because I said it was passive. 4.2, use your knowledge of transport across, actually I should go through this first. A scientist measured the rate of uptake of a monoglyceride and a monosaccharide, one of those little sugars that you learnt about right at the start of AS, by epithelial cells of the small intestine of mice. A monoglyceride is a molecule of glycerol with 1 fatty acid attached. Theoretically, you should already know that. She did this for different concentrations of monoglyceride and monosaccharide. Okay, all right. Use your knowledge of transport across membranes to explain the shape of the curve in figure 1 for the uptake of monoglycerides between concentrations. So, between concentrations A and B. Well, between concentrations A and B, as the external concentration increases, so does the rate of uptake. It is positive correlation. So what did I write? I really can't remember. Yes, and therefore, so yes. here we are, you're explaining. Notice that you are explaining the shape of the curve. Notice what I did just then was describing it. I said, oh, as the external concentration increased, so did the rate of uptake. Well, that's a description, not an explanation. So this is diffusion because, and I'm explaining why, the evidence from the graph. Between C and D, the carrier proteins were saturated and therefore uptake plateaued, because I've worked out in my head already that this is facilitated diffusion. The shape of this graph shows facilitated diffusion. And that is what I have concluded here. Both of these are diffusion. But this one, the monosaccharide, is facilitated diffusion, and the monoglyceride is actually simple diffusion. So here is the mark scheme here for 4.2, and here is the extra guidance, and therefore this is where I got the mark points. So movement through carrier proteins or facilitated diffusion. In fact, I probably got the mark point 1 here, actually, because I said that carrier proteins were involved. I just realized that I didn't place that tick quite accurately. And here I got mark point 2 because the rate is proportional to the rate of the concentration increase. And finally, here was mark point 3, that those carrier proteins were saturated, and therefore they limited how much was taken up. Figure 1 is evidence for monoglycerides being lipid-soluble molecules. Well, it's because simple diffusion is happening, and it is a straight line. There is no plateauing off. As the external concentration increases, the rate of uptake also increases, because as the external concentration increases, the concentration gradient between the outside and the inside of the cell steepens, and therefore the rate increases. So, and I said they move by simple diffusion because they are lipid soluble. Here is the mark point, here is the guidance, and I got mark point 1 here because I said there was no plateauing off, and I got mark point 2 because I pointed out that they are going straight through. I said simple diffusion, and that means that they are going straight through the phospholipid bilayer. They're not using a carrier protein. Kelsey, could you say anything about intrinsic proteins, since osmosis uses aquaporins? Let me go back to yes, you could say that. I'm going to be honest with you, Kelsey. Talking about aquaporins isn't something that is generally done very much. It wouldn't be emphasized at AS. They are absolutely talked about in year 13. I bet it's not on the mark scheme though, is it? No, both of them. No, see, there's the bummer. Because you were thinking in your mind about facilitated diffusion, and you were thinking about the situation where osmosis is using aquaporins, you rightly said that in those situations, they both use a type of channel protein. You talked about intrinsic proteins. I would certainly have called them both a channel protein. It isn't the most obvious thing to say. It isn't inaccurate in my view, but I'm going to be honest with you, it's not on the flipping mark scheme, is it? And if I'm really honest, because I didn't mark, the AS Biology paper in 2018, I don't know how that answer would have been handled by examiners, because if they had included it, I would have expected there to be a note on the mark scheme, on the final mark scheme. That's as good as I can do, Kelsey. I hope that's useful. Right, let's just zip down through this and move on to the next question. A student, new question, new topic. Student prepared a stained squash of cells from the tip of an onion root and observed it using an optical microscope. During the preparation of the slide, he cut the first 5 millimeters from the tip of an onion root and placed it on the glass slide. He covered this tip with a drop of stain solution and a cover slip. He warmed the glass slide. He pressed down firmly on the cover slip, and he identified and counted nuclei in different stages of the cell cycle. Explain why the student used only the first 5 millimeters from the tip of an onion root. And I am noticing that there are 2 marks here for both parts of this question. Therefore, each part is only 1 mark. We're not expecting a full essay as an answer. So I think the thing that I wrote obviously here was that it is only in the very tip of the root where mitosis takes place. And the whole point of this was to see the different stages of the cell cycle. In other words, the stages of mitosis. So because it is here where mitosis occurs in the root, it is only there really. And why did he press down firmly on the cover slip? Classic answer here, to make the specimen thin enough for light to go through. In other words, you're kind of aiming for the specimen to be only 1 or 2 cells thick. So here is the mark scheme. Here are the additional notes. And therefore, I got mark point 1 here. And I got mark point 2 here. Notice that this is like a very classic question. You know, why does the specimen have to be thin, or how thin does the specimen have to be? You know, describe and explain how thin the specimen has to be. Has to be 1 or 2 cells thick so that light can pass through it. If light doesn't go through a specimen for an optical microscope and a light microscope, you won't see anything when you look down the eyepiece. Figure 2 shows the cells the student saw in one field of view. He used, now this is slightly weird because that field of view is rectangular. I've never seen a rectangular field of view when I've looked down a microscope. I've only ever seen a circular one. But anyway, we will ignore that for the moment. He used this field of view to calculate the length of time these onion cells spent in anaphase of mitosis. Okay, all right. Scientists have found the mean length of time spent by onion cells in anaphase of mitosis is 105 minutes. The mean length of time, average length of time. They also found that the cell cycles of cells in the onion root, shown in figure 2, take 1,080 minutes. The whole cell cycle. That whole, you know, interphase, G1, synthesis, G2, and importantly, mitosis, and right at the end, cytokinesis. The whole cell cycle. From going from one little baby cell, that baby cell growing up into a big cell, and then getting to the stage where it divides into 2 new daughter cells. 32 whole cells are shown in figure 2. I'm going to take them at their word. Use this information and figure 2 to calculate the length of time the cells of this onion root are in anaphase, and then calculate the percentage difference between your answer and the mean length of time found by the scientists, 105 minutes. So this is like a snapshot photograph. It's like taking a snapshot aerial photograph of people at a concert and going, you know, how many people have their hands in the air? And because it's a snapshot, you can then estimate the length of time that people had their hands in the air. I'm sorry, that's not a great analogy, but because it's a snapshot, you can make an assumption. Oh, let's think about this another way. This shows a whole load of cells. Intuitively, just looking at these cells, most of them are in interphase. They are just showing nuclei, which are not, you can't see any chromosomes visible, and therefore we're pretty flipping sure that the cells with no chromosomes, well, we're absolutely sure that the cells with no chromosomes visible are in interphase. And therefore, intuitively, just by looking at this photograph, this snapshot of a moment in time, you would kind of say, well, I would say that most of the time cells are in interphase. Does that kind of make sense? And actually, you can extrapolate that kind of gut intuition into a mathematical expression. Because if you know the whole length of the cell cycle, the fraction of the cells that are in a particular stage, in this case anaphase, times by the whole length of the cell cycle will give you the length of time that the cells are in anaphase. So in figure 3 cells are in anaphase. This one here, and this one here. So the fraction is 3 out of 32 times by 1080, which equals 101.25 minutes. That's the first part of the answer. You've then got to calculate the percentage difference. Now, the percentage difference, you can either calculate the percentage decrease or the percentage increase. Either is valid. I chose to calculate the percentage decrease. So the percentage decrease is going from a larger number to a smaller number, working out the difference as a proportion of that larger number. So it's 105 minus 101.25 divided by the original larger number, 105, times by 100, which gave me 3.57%. Here is the answer, which gave you 2 marks. Here are the notes. Now, notice these larger numbers, so 3.57 and 3.6, are this number. These numbers here are if you chose to calculate the percentage increase. And that, of course, instead would be going from a smaller number up to a larger number, working out the difference, but then working it out as a fraction of the smaller number. So therefore, it's 105 minus 101.25 divided by that smaller number, original number, times by 100, which gives you a slightly, well, gives you a different figure. There we go. I got both of the marks. Tick the name given to the division of cytoplasm during the cell cycle. It is cytokinesis. Over 90% of students got this right. So here we go. Describe and explain what the student should have done when counting cells to make sure that the mitotic index he obtained for this root tip was accurate. 2 marks, describe and explain. There'll be a mark for both. So I said that he should count a large number of cells using a large number of fields of view to ensure that his sample was representative of the whole root tip. This word here is a really useful word to use in answers when you're talking about taking samples of the whole. So just a good hint there. So description and explanation. So I got mark point 1 and mark point 2. Notice that you could have done 3 and 4 or 5 and 6, but these were the ones that seemed most, this was the 2 that seemed most obvious to me. A scientist treated growing tips of onion roots with a chemical that stops roots growing. After 24 hours, he prepared a stained squash of these root tips. And here is a drawing of the chromosomes of a single cell. This cell was typical of other cells in anaphase of these root tips. Now, this is tricky to decipher, I think. But what I think we're looking at is that these are all the chromosomes that are at the center of the cell during metaphase. And some of them, look at these ones, all of these are this V shaped. These are being split apart during anaphase, but you've got a whole load here that have stayed still. That's what I think we are looking at, because there are quite a number of straight chromosomes. Use all of this information to suggest how the chemical stops the growth of roots. So I have assumed that the spindle fibers could not attach to the centromeres, therefore the sister chromatids, we're talking mitosis, not meiosis, so the sister chromatids could not be pulled apart to opposite poles of the cell, and therefore those chromosomes could not be divided equally between the 2 new cells. And as a result, cell division couldn't carry on, and therefore growth stopped. And this is pretty much what I wrote. Here is the mark scheme, and here are the additional notes. And therefore, this is where I got my marks. I didn't get mark point 1. I got mark point 2 because I kind of jumped straight in and kind of said that the spindle fibers wouldn't attach. And I also said that the sister chromatids couldn't be separated. And I think that was the final mark. I only got 2 out of the 3 marks because I didn't go on to say that final clinching point that no new cells are added. And I didn't say categorically that it stopped cell division. I said that cell division wasn't successful. And I'm going to be honest, I was kind of envisaging the 2 cells being produced with one cell having potentially either no chromosomes in it or very few, and the other cell having more. You know, I didn't envisage it quite the way that they wanted me to. So I didn't get them on. New question. Why do you times? Oh, why do I times by 1080? Because if you go back to that question, the time for the whole cell cycle was 1080 minutes. So because the number of cells going on in anaphase in that snapshot, in that photograph, was 3 out of the total of 32 cells, you can assume that that is the fraction of the whole cell cycle that takes that is the duration of the time that anaphase occurs. So if you remember when we looked at that picture, most of the cells were in interphase, and just intuitively you would have just kind of said, hey, it looks to me like most of the time the cells are in interphase because there aren't very many cells going through mitosis. You can take that logic to a mathematical expression, because the estimate of the time taken for any particular phase is the fraction of the cells in that phase times by the time taken for the whole of the cell cycle. I hope that makes sense. Okay, question 6. Under the correct conditions, new roots grow from the cut end of a plant stem. A scientist investigated the effect of substance X on the growth of new roots. She used a ringing experiment to investigate the movement of substance X in stems taken from lemon plants. Okay, she cut out a length of stem from each plant. She then put a small block of agar on the top of each stem. Some agar blocks contain substance X. D, the agar block does not contain substance X, and the stem is intact. E, the agar block contains substance X, the stem is intact. F, the agar block contains substance X and the phloem has been removed. G contains, the agar contains substance X and part of the phloem is cooled right down to 4 degrees C. She grew the stems in the same environmental conditions for 6 weeks and then found the number of roots per length of stem. Roots grew out of the other end of the stem where the agar blocks were placed. So they grow down the bottom here. Okay, and here is the mean number of roots per length of stem. Treatment D is a control. Explain how the measurement obtained from this control is used by the scientists. Well, it's that part of the experiment. A control is used to compare with all the other parts of the experiment, because a control is going to show you the effect of the active ingredient in the experiment. In other words, substance X. So this is what I said. It is used to compare with E, F, and G. The comparison shows the effect of X on root growth. Here is the mark scheme. And so I got both marks. I didn't say as a baseline, but I said it's used to compare the effect of the other treatments. So, and I've named them here. And then I got mark point 2 here. Notice that a control is very different to a control variable. A control variable is one of those things that you keep the same so that the experiment is a fair test. In other words, your experiment and therefore your data is valid. An example of a control variable in this situation would be the length of the stem used in the experiment, or the fact that you use the same species of plant, for example. Using figure 4 and table 3, what can you conclude from treatments D and E about root growth? D and E. Okay, so here you have got 5 roots in D, and E you have got 11. So you could conclude that substance E moved down the stem from the top to the bottom and it increased root growth, which I'm hoping is what I put. Yeah. Oh, I added something else because this, notice that I wrote these 2 things, and to be quite frank, it's 3 marks. I've really only written 2 statements. So then I was kind of going, okay, what else can I write? Oh, it moves via the phloem, as this is the only 2-way route of transport. So there was me trying to say, the xylem, this is the direction of the xylem. I don't think I expressed this very well, by the way. This is the direction of the xylem. And of course, the phloem can move stuff in both directions. So it's the only way in which the stuff could have moved down the stem. That's what I was getting at. Can't remember what the mark scheme was. Here are the notes. So I got mark point 2 and I got mark point 3 because it increased root growth. I didn't get mark point 1, so I only got 2 out of 3 marks. 6.3, the mass flow hypothesis. Will I be doing the AQA AS Biology 2019 papers? No, I won't, and I won't be doing the 2020 papers either, because both of those years are still secure papers, which teachers in school absolutely bank on keeping secure so that they can use them as mocks at a later date. So I don't go through any papers publicly which are still secure papers. Teachers in school would rightly want to shoot me if I did. Would love to see these walkthroughs for GCSEs. Yes. Oh. Okay. All right. Habib, I do understand. I do concentrate on A level at the moment. Almost certainly, I'm going to say to you, if there was stuff on the paper that you hadn't heard about, it was almost certainly because you were expected to apply your knowledge to a new situation. In other words, you would have, the only way you would have got marks is by using the knowledge that you had learnt at GCSE, and it had been applied to a situation that was unfamiliar to you, and that is quite normal. Yes, because I know that I don't think that symbiosis, for example, is in your syllabus, but you wouldn't have needed, almost certainly, you wouldn't have needed to know what symbiosis is. There would have been some aspect of the question that you needed to apply your knowledge. I'm certainly hoping so. Anyway, back to this question. Right, mass flow hypothesis is used to explain the movement of substances through phloem. Evaluate, oh my goodness, evaluate means pros and cons, advantages and disadvantages. Evaluate whether the information from this investigation supports the hypothesis. Do not consider statistical analysis in the answer. So really kindly, they're absolutely telling you what isn't in the mark scheme. So here, so what are the things that are for the mass? So here we go, evaluate, plus and minuses. It's the first thing I think of when I see the word evaluate. It's either pros and cons, fors and against, advantages, disadvantages. So here we are, what information from this investigation supports the mass flow hypothesis? Well, 4 is that the phloem is involved, as F shows root growth equals control D when the phloem is removed. So in other words, so if the phloem is removed, the amount of root growth equals what happens in the control, and therefore it strongly implies that the phloem is involved. It also shows that respiration is involved, and we know from our knowledge of translocation that respiration is involved, energy is required, as G shows that the root growth equals the control, D, because 4 degrees C will slow respiration significantly. Things that are against: roots still grow in F when the phloem is removed. And also, now this was me really, I'm going to be honest, really racking my brains. And I don't think this is obvious at all, if I'm honest, because it would require you to remember the ringing experiments, which I think is not a major part of the syllabus. So you would have expected to have had a bulge here, because of course stuff would have been moving down here. It would have been removed from this part of the phloem, but it would have collected here. So this is what I wrote. Here is the mark scheme, and here is where I got the marks. So I got mark point 1 here. I got mark point 2 here. And I got mark point 6. I don't think I got, oh, I did get, oh, right, yes, and I got mark point 9. So I did end up getting all 4 marks. I think 2 or 3 marks would have been a perfectly good answer for most students. Question 7, what is digestion? Right, this is a definition straight from GCSE. It is, now I wanted to write, it is the breakdown of large insoluble molecules to small soluble ones, but then I suddenly realized I don't want to use the word breakdown, I certainly don't want to use the word digestion, I should be using the word hydrolysis because that is an A-level term. So that is what I put. Here's the mark scheme, and therefore I got the 2 marks. One species of fungus digests cellulose using 2 types of enzyme, endocellulases and exocellulases. Ooh, that sounds familiar, doesn't it? Endopeptidases and exopeptidases. And as I suspected, endocellulases act in the middle of the cellulose molecule and exocellulases act at the ends. Endocellulases and exocellulases act at different places on cellulose molecules. Suggest why. Here is what I put. I wrote this answer first. Why do they act at different places? Because, oh, actually, I've just realized that was really, right, so it increases. This was a kind of standard answer for endo and exopeptidases. Why are they both used? And I only realized that this was the wrong answer because in a couple of questions' time, this is very definitely the right answer. So I then went back and looked at the question again. Why do the enzymes act at different places on the cellulose molecule? And it was then that I realized that this is a flipping enzyme question. It's all about shapes of the active site. So here's the tip. If you are talking about why a particular enzyme works on a substrate and doesn't, always talk about the shape of the active site or the tertiary structure of the active site. Always emphasize that, which is what I did here. And therefore, to kind of nail the point, therefore, so it's the active site is complementary to different parts of the cellulose molecule. These are really specific enzymes. Hope that makes sense. Here is the mark scheme, and therefore I did just about manage to scrape the marks. If anyone, yeah, please don't DM him, actually, because you would be cheating. And Abaydullah, you shouldn't have the flipping papers, but anyway, really, anyway, okay, moving on. Scientists prepared the following mixtures. These ones here. Oh, this one has both. The mixtures had identical total volumes. She determined the mass of the cellulose remaining after 48 hours. Notice that this is where you've got to read questions carefully. This is the mass of cellulose remaining, not the mass of cellulose that has been digested. Use information from table 4 to calculate the rate of digestion of cellulose when both enzymes are present. We're using this figure here. Give your answers in grams per minute and in standard form. I'm going to come back and, remind myself of this at the end of the question. So we've started off with 15 grams. We have ended up with 9.2. So the amount digested is 15 minus 9.2 over a 48-hour period. So that's how much was digested in 48 hours. Therefore, if you want it per minute, you've got to divide 5.8 by 48 and then divide that number by 60 to get it per minute, which gives you this number on your calculator. And they want it in standard form. So 1, 2, 3, it's 2.01 times 10 to the minus 3. There we go. So I got the 2 marks. The scientists used the same concentration of endocellulose and exocellulase, endocellulase and exocellulase in the mixtures. The rate of digestion of cellulose is greatest when both enzymes are present. This is the question that requires the answer about, oh, that's because endocellulase digest in the middle and exocellulases then digest the terminal ends. So this is what I wrote, and I got the 2 marks, fortunately. Here, the scientist could have expressed her results as the percentage loss in mass of cellulose. Percentage loss. So this is, we've talked about this already. The equation for percentage loss is the initial minus the final mass divided by, because we're talking about percentage loss, it's divided by that higher initial value, divided by the initial value times by 100. So there we go. All the other connotations here or combinations here. 8.1, we're getting there slowly but surely. A student used a dilution series to investigate the number of cells present in a liquid culture of bacteria. Describe how he made a 1 in 10 dilution, and then how he used this, notice it's in bold, how he used that 1 in 10 dilution to make a 1 in 1,000 dilution of the original culture, liquid culture of the bacteria. So if you want 1 in 10, you have got to take 1 part of the original culture and dilute it with 9 parts of, well, either water or just the nutrient broth. Doesn't matter which. You're basically diluting it 1 into a total of 10. Now, I wrote it as this. Take 1 centimeters cubed of the bacterial culture and dilute it in 9 centimeters cubed of nutrient broth, because you would end up with 1 centimeter cubed in a total of 10 centimeters cubed. It would be a 1 in 10 dilution. If you took a centimeter cubed of this dilution, it would have 1/10 of the number of bacteria in it. Yeah, hopefully that makes sense. And notice, shake well, yeah, because you need those bacteria to be spread out evenly throughout your dilution. How would I use this dilution then to make a 1 in 1,000 dilution? Well, now we've done 1 in 10, but we need to dilute it again by another factor of 100, because 10 times 100 equals 1,000. So now I've got to make a 1 in 100 dilution of this dilution. 1 in 1,000 dilution, take 1 centimeters cubed of the 1 in 10 dilution and dilute it in 99 centimeters cubed of nutrient broth. We're now making that 1 in 100 of a dilution. And 1 in 10 times 1 in 100 is 1 in 1,000. Okay, so here is the mark scheme, and this is where I got the marks. Notice there was a mark for just shake well, rather handily. Okay. Using an optical microscope, the student determined that there were 15 cells in 0.04 of a millimetre cubed. Oh my word. Teeny tiny amount of liquid of the 1 in 1,000 dilution of the culture. Here am I visualize this. Here am I have got a fraction of a centimetre cubed. Calculate, sorry, let me just finish this. Calculate the number of cells in a centimetre cubed of undiluted liquid culture. So we have got a fraction of a centimetre cubed of the 1 in 1,000 dilution, and they want to know how many cells there would be in a centimetre cubed of undiluted culture, in the original culture. Potentially quite scary. So, first of all, I scale up, I kind of go, right, okay, let's break this down. How many cells would there be in a centimetre cubed of diluted culture? So this is what I did first. If there's 15 in 0.0, that decimal point's hardly there, 0.004 millimetres cubed, therefore, this is the scaling up that I need to do to work out how much there is in a centimetre cubed. And it's 1,000 over 0.004 because a centimetre cubed is 10 millimetres by 10 millimetres by 10 millimetres, which is 1,000 millimetres cubed. So a centimetre cubed is 1,000 millimetre cubed. So therefore, if we've only got 0.004 millimetres cubed, this is how much we've got to scale up this number 15 to get how many bacteria there are in a centimetre cubed of diluted broth. Therefore, in undiluted culture, it is this times by 1,000 because you've diluted it by 1 in 1,000. Does that make sense? And therefore, the number is, it was 3.75, and then after 300, it's 375 million. So in other words, 375 and then 6 zeros. And that, of course, in standard form is 3.75 times 10 to the power of 9. Hopefully that makes sense. Okay, all right. Okay, oh my goodness. Will we ever get a question like that? Well, Sarah, you'll always be given the application. Sorry, I just realised the whole load of questions going on, so let's cover some of them. Sarah, will there always be application questions? I mean, even some of these ones that we've gone through, you've talked about things that are in unfamiliar scenarios for you, where you've got to apply your knowledge to the unfamiliar scenario. Don't be scared by it too much. Okay. Well, we've got live stream. Johnny, all the live streams that get saved onto YouTube. So yeah, absolutely, you can watch it later. Thank you, Johnny. Oh, you're really sweet, Johnny. Thank you. Yeah. Thank you. Okay, a bit of chat between you guys. Loving the conversation. Thank you very much. I really appreciate your confidence in me, Johnny. I appreciate that very much. Okay, brilliant. Thank you. Not so many questions. Just you guys bigging me up. Lovely. Right, I'm going to carry on now. The student looked at cells in a 1 in 10 dilution. Remember, visualize this. This is the one where he's only diluted it 10 times. Yeah, not 1,000 times, just 10 times. He decided not to use this dilution to determine the number of cells in the undiluted liquid culture. Well, the most obvious reason to me is that he's looking down his microscope and there's too many bogging bacteria to count, because he hasn't diluted the culture enough times. You know, there are just hundreds or thousands of them in one field of view. Just not possible to count accurately. You're never going to keep track of the little blighters, are you? Okay, all right. So suggest an explanation for the student's decision. So this is, oh, sorry, I've just realised I didn't mark that one. Boring, boring, boring. Yeah, got the marks. Okay. This is what I wrote. And I kind of went, oh my goodness, I've hardly written anything. But I seriously kind of went, there would have been too many cells to count accurately. And I kind of thought, can I add any more blurb to this to make it sound more intelligent, or to add more detail? And for a few seconds, I just thought, I don't think I can. I think that may be enough for the 2 marks. And I literally kind of went, oh, well, if it isn't, it isn't. And I moved on. One of those situations where don't be scared if you don't fill up all the flipping lines, but you're pretty confident you've written as much as you possibly can. And I'm somebody that does like to write. I will write a little bit more detail if it only takes a few more words to be as sure as I can be that I'm going to bag the marks. I didn't in this case. And here's the mark scheme. How funny is that? Yeah, you know, but hopefully, and notice it's because I was confident. I knew what I was writing. I knew that there would have been way too many cells. And I knew that a key word, and this was my suspicion, that the key word was, in my view, accurately. He couldn't literally keep track of what he was trying to count in the field of view. Sorry, is it he or she? I can't remember anyway. Yeah, so yeah, there we go, moving on. 8.4. On some farms, animals are routinely given antibiotics in their food. Scientists investigated whether these farm animals had antibiotic resistant bacteria in their intestines. They tested the bacteria for resistance to 2 antibiotics, tetracycline and streptomycin. Notice we're told they are routinely given antibiotics in their food. All right, they're just, you know, just chucked in there with the feed stuff. So 29% are tetracycline resistant, 13% are streptomycin resistant. Suggest and explain one reason. Notice, suggest and explain. So give the reason and then explain it. Why bacteria resistant to tetracycline are more common than bacteria resistant to streptomycin in these farm animals. And my logic was that they had been fed the tetracycline for a longer period of time, and therefore there had been enough time for the resistance to build up. What had I written? Been used for longer than streptomycin. Therefore, oh yeah, I did write it a little bit more intelligently. Therefore, more generations of bacteria would have passed down the tetracycline resistance allele. That's what I wrote. Here's the mark scheme. So I got, and here's the guidance, I got mark 0.5, but I missed mark 0.6, which when I look at the mark scheme, I kind of go, oh, darn. Yes, because of course, if they talked about it in terms of the number of mutations building up, whereas I was kind of thinking about it in the more bacteria were being reproduced that we're going to end up with the allele. So I didn't get that second mark. hey ho, I'm not going to worry about it. In recent years, these farm animals have not been given tetracycline in their food. Despite this, the percentage of bacteria resistant to tetracycline has remained constant. Suggest one reason why. Well, just because they haven't been given tetracycline in their food, those alleles are just going to be passed down each generation of bacteria. This is, oh, right. So this is what I put, kind of thought, well, they haven't, these tetracycline resistant bacteria haven't been killed. And therefore, the numbers aren't going to reduce. And I put in the word selectively killed. Here's the mark scheme. And I can't remember, I wasn't sure whether I gave myself, I did give myself the mark because I put in the word selectively. So I was going for this one here, no selection against resistant bacteria. I named what would have selected. I'm not absolutely sure I'd have got the mark, I'm going to be honest. I don't know. I think probably I would because I did talk about selection, but yeah, a bit tricky. Hey, you can not give me that mark if you want to. 9.1. Can I just say the number? I think the number of marks available on this exam were, I think it was 100. Have I got that right? And I might go back and check this. I'll check the grey boundaries in a minute. Compare and contrast the DNA in eukaryotic cells with the DNA in prokaryotic cells. I read the examiner's report for this question. There was an awful lot of confusion by students about the difference between prokaryotic and eukaryotic. A lot of students thought that the DNA was fundamentally different in the structure between eukaryotic and prokaryotic. And yeah, there was a lot of confusion here. So this is what I wrote. Here we go, big full. So the first thing I said was that both are double helices made of 2 polynucleotide strands. Can I just emphasize the DNA in bacteria, in prokaryotes, is fundamentally the same. It is the same molecule, not fundamentally. It is the same molecule as is in eukaryotes. It's that double helix made up of 2 polynucleotides, so made up of loads of made up of the same nucleotides. Phosphate, deoxyribose sugar, and a nitrogenous base, adenine, thymine, cytosine, or guanine. Yeah, so that is the same. Eukaryotic DNA is longer than prokaryotic DNA, way more of it than eukaryotic. DNA is linear in eukaryotes, whereas it's circular in prokaryotes. Eukaryotic DNA is associated with histones, whereas it isn't in prokaryotes. And I kind of finished here. I'm going to be honest, I kind of did the full stop here, except it's not even there. And then I kind of thought, I can't think of anything else to write. What have I missed out? And then I realized that I'd started down the line of, because it says, let me just point out, it says compare and contrast. Contrast is differences. Compare is just talk about, it's kind of saying, talk about the things that are the same, but also talk about the things that are different. Yeah, so it is it's both of those things. And therefore I suddenly realized I actually haven't talked enough about the fact of all the things that are the same. I realized that I'd subconsciously, once I'd done this little sentence at the top here, I then concentrated on the contrasts. And I hadn't done enough about the things that were the same. So therefore, I kind of went back and said, both are made up of nucleotides comprising a phosphate group, deoxyribose sugar, and a nitrogenous base. And the two polynucleotide strands are held together by hydrogen bonds. And, here I really did feel like I was scraping the barrel, I'm going to be honest. And also to say that the phosphodiester bonds holding those sugar phosphate backbones together are also the same. Here's the mark scheme. Here's a little bit of guidance. So mark point 1 here, mark point 2 here, phosphodiester bonds. Mark point 3, didn't even think of, not even on my radar. Mark point 4, I got up here, longer. Mark point 5, oh my goodness, what a dipstick. Forgot this one completely. Anyway, mark point 6, got mark point 6, and I got mark point 7. So I got the 5 marks. If it had been a 6-mark question, I clearly wouldn't have got the full marks because I missed out something that I really should have remembered. I wouldn't have, I'm going to be honest. I know this is true. I know mark point 3 is true. It didn't even cross my mind when answering this question. I should have remembered flipping introns and exons. Anyway, not to worry. Final question, here we go. Hemoglobins are chemically similar molecules found in many species. Differences in the primary structure of hemoglobin molecules can provide evidence of phylogenetic relationships between species. Explain how. This is what I wrote. Oh my goodness, what a load of stuff. Right. The first thing I did was that I noticed that they hadn't defined primary structure. So I thought, right, let's kick off. Let's define primary structure, and let's also make sure that we really nail what phylogenetic relationships mean. So those were the two things that I kind of thought, I definitely want to do that. So primary structure is the order of the amino acids in a protein. The sequence of amino acids in different hemoglobins can be identified. You can work out the sequence. The more similar the sequence order, the more closely related the two species are. Here we go. Now I'm going to nail what the phylogenetic relationship thing means. This means that those two species, have a more recent common ancestor. Here's my tip to you. If a question talks about phylogenetic relationships, use this phrase in your answer. More recent common ancestor than other pairs of species. Yeah, hopefully that sentence makes sense. Now, I'm going to be honest, I got to this point here and I kind of thought differences in the primary structure of hemoglobin can provide evidence for phylogenetic. How can differences provide evidence for? Well, I'm going to be honest. At this point, I thought I'd written pretty much all I could write. How do the differences provide evidence for the evolutionary relationships, phylogenetic relationships between species? And I knew I hadn't written enough for 5 marks. And so I felt here that I was blurbing, absolutely scraping the barrel. Because then I kind of thought, do you know, all I can think of is that I need to kind of go back and talk about DNA. I really wasn't convinced by this. Primary structure of a protein is determined by the base sequence on the DNA, each base triplet coding for an amino acid. Mutations, changes in the base sequence of the DNA, occur over time, which means different amino acids are coded for and the primary sequence changes. I felt that this bit here was me really answering the questions, because I read the question as, how do differences in the primary structure of the protein provide evidence for? How do those differences provide evidence for? Well, the more differences there are, the or the least differences there are, the more recent the most recent common ancestor is. Yeah, that was, I felt, was answering the question. Here's the mark scheme. So these are the marks that I got. I got mark 0.1, mutations change the base sequence. I got mark 0.2, saying that that causes a change in the amino acid sequence. I didn't get mark 0.3. I didn't say, even though I said occur over time, I didn't say build up over time. It is different. I didn't give myself the mark. I got mark, I didn't do mark 0.5, mark 0.4. I did get mark 0.5 because I nailed the difference between common ancestor. Notice, if I had just written that first mark, that first bit, I would have only got 1 mark out of 5. I ended up with 3 marks out of 5. And I must admit, when I saw the mark scheme and read the mark scheme, I thought, flipping heck, that's a bit crap for a teacher. So I then went and looked at the examiner's report. Here's the examiner's report for this bit of the mark scheme. In 9.2, it was evidence that students had difficulty with this topic, and many answers, over half, gained no credit. Over half the students got 0 out of 5. I think that's significant. That says it was flipping hard. Some students were diverted because the question referred to hemoglobin and went into long detail about levels of protein structure and physiological adaptations to different oxygen environments. Yeah, fair enough. They went off on one. They weren't answering the question. Those who discussed evolutionary relationships often gave vague answers that referred to sharing a common ancestor or being related, rather than use ideas like the more differences would indicate a more distant relationship and an earlier common ancestor. I at least nailed mark 0.5. Okay. Very few students considered what caused the differences in the primary structure of the hemoglobin. Consequently, little mention was made of mutation. I'm going to suggest that there are 2 very different ways of reading this question. Either emphasizing how do the differences in the primary structure provide evidence, explain how the evidence is provided, in which case this, well, particularly this bit answers the question, but clearly it's only 1 mark, yeah, or else say, explain how the differences in the primary structure come about and therefore are evidence of a phylogenetic relationship. Can you see there's a really different way of reading the question? Which is why I think very few students considered what caused the differences in the primary structure of hemoglobin. Consequently, little mention was made of mutation, which is the process underpinning species change in evolution. Absolutely it is. Absolutely it is. It's not the way I read the question. Few considered why these differences accumulated over time. Okay, many answers achieved 2 marks, many. I would suggest that that was probably 70+%. And rarely more than 3 marks. The mean mark on this question was less than half out of 5. That, I think that tells you that an awful lot of students tried answering the question kind of in the way that I did in this first bit here, and then simply didn't know what else to say. A bit like me. And I felt like I was dredging the bottom of the barrel, but ended up getting another couple of marks. Really hard question. So notice that if you if you completely screw up a question and you kind of go, hey, I really thought I knew my stuff. Oh my goodness, what went wrong? And the mark scheme doesn't help you. Absolutely go and look at the examiner's report, because sometimes that absolutely puts in context why you found the question difficult. And it also gives you an idea as to how worried you should be about losing marks on that question. I read that examiner's report and I kind of went, oh, do you know, whatever. I'm not worried. Let me quickly go back. I just want to remind myself, silly old me. Right, let me quickly go back because I want to just be sure. Right, the maximum number mark on this paper is 75. So I'm pretty sure that the grade for an A across the two papers was, I hope I'm right in saying this, was 80 out of 150. I dropped, I think I added up my marks, I dropped less than 10 marks on this paper. So, you know, even though I felt like I dropped quite a large number of marks on this paper and was, you know, kind of thinking, flipping heck, I'm a teacher. Actually, clearly, I'm in very high A-star territory, which is kind of comforting, seeing as I teach the flipping subject. But, you know, I'm really emphasizing, you know, whatever grade you are aiming for, clearly you want to be sure that you're there or thereabout hitting the number of marks that you need for your grade. But notice that there are some questions that some answers or some mark points, let's be realistic, there are mark points that you can just kind of go, oh, do you know, whatever. I would never have written that. I would never have thought about it. And frankly, I'm not going to worry about it. This is where, when you go through an exam paper, you really want to be self-critical, because you need to be able to identify the marks that you should be getting. I should have got that intron difference between prokaryotic and eukaryotic DNA. If I'd lost a mark because of that, I would have rightly been cross with myself. Yeah, that was a wasted mark. If I'd made a stupid mistake in the maths, that would have been a wasted mark. I note to self, I need to be more careful. You know, I need to read questions more carefully, whatever. But if you look at a mark scheme, and particularly if you look at an examiner's report and go, oh, flipping heck, I would have never have got the marks, write them off. Write them off. Unless, and here you've got to be self-critical, if you haven't got enough marks to get the grade that you need to identify those marks where it is a lack of knowledge that is stopping you getting the marks. Because if it is a lack of knowledge that is stopping you getting the marks, you just didn't know the answer, then you have got to work out how you are going to revise more effectively. Because the bottom line is, if you don't know the content of your biology A level, you cannot get very many marks on the paper. All right. So it is, and I talk a lot about that on my, oh my goodness, freebie video on my website, which is www.biologycarol.co.uk. There's a freebie video on that website which talks about how to get your grade up to an A star. And I go through all of those things about analysing your own papers, being critical about why you lost marks, because you can only change the way you work if you understand why you are losing the marks. And then in that video, I also talk about things like how to revise effectively, how to attack things like drawing conclusion questions, evaluating questions, all of those kinds of things. So do go on the website and have a look, and hopefully you will end up agreeing with the lovely Johnny and Glamour Vibes, love the name, about the stuff on my website being stupidly good value for money. Basically me teaching the whole of the A-level biology course for the cost of 2 private tuition lessons. And I hope that has been useful. But I am going to love you and leave you now because I desperately need a cup of tea. My voice is going. And I will see you in, I think 4 weeks' time. I'm away for the next 3 weeks. Sorry about that, guys, but hopefully that has been useful. Thank you, Glamour Vibes. 81 was the A. Of course, you don't get A stars at A level. So I hope that was all useful. Let's love you all. If you've still got exams coming up, go for it, and I will see you in a month's time. Okay. Bye-bye.